So for this slide, you will learn in detail about frequency of action potential and also all or nothing law. Okay, so what is the frequency of action potential? So these two graphs is all about frequency of action potential. So this is the membrane potential showing you the action potential that has been produced starting -70 millivolt, threshold level is being achieved, and spike to +30. Okay, so this we can call it as a weak frequency of action potential. This is high frequency of action potential. The production of action potential with different kind of frequencies, either weak or high, it's actually depend on the stimulus strength. So for this one, you can see weak stimulus strength just on border at threshold level, reach the threshold level, but on border, so we can say it is weak stimulus strength compared to this one, bigger and larger scale, way to fall from the threshold level more than -55. Okay, so that's why it can produce higher frequency of action potentials. So from this graph, okay, any kind of stimulus strength that can't reach the threshold level at -55 millivolt, that will be null data of action potential. Action potential would never produced yang F kosong. And this is called all or nothing law. All mean it just only that stimulus that can reach threshold level on point or more than that. Those can produce action potential. For any stimulus that weaker than that of -55 millivolt, no action potential will be produced. So it indicates no action potential will produce. That's why nama dia all or nothing law. Dia tak ada nak kata tengah-tengah. Nak kata tiba-tiba dekat bahagian ni jadi pendek sikit ke, tak ada. Sama ada tak ada langsung ataupun all. All mean all complete amplitude starting -70 shoot to +30. All means tak kira lah weak stimulus, even though weak, dia still capai threshold level, ataupun strong stimulus, amplitude size is just the same. That can differ. These two kind of action potential is based on their stimulus strength. Okay, so from this graph, we can conclude that all or nothing law. All means that capai je threshold level on point or more than that, there will be production of AP, but if not achieved, will be null. Tidak ada AP will be produced. AP is action potential lah. Okay, so this is we call the all or nothing law response. So actually frequency of action potential here, the explanation can be made. Information in axons is coded in the frequency of the action potential. Okay, messages is encoded in the frequency of the action potentials. Weak stimulus above threshold level, this one, weak stimulus above threshold level, produce fewer action potentials, mean give out a weak response. Okay, weak intensity of response, whereby stronger stimulus are way too far, I mean like a larger scale. Stronger stimulus produce a greater frequency of action potentials. As the intensity of stimulation increase within limits, dia ada limit juga, okay, more action potential are triggered off. Okay, I come again. Semakin tinggi intensity of stimulation, more action potential are triggered off. Okay, but this is actually under limit, within specific limits. So when our nerve cell is being strikes on this kind of stimulus, this means that our nerve cells and also our axon is said to be firing. But how does the firing has to be controlled not to be over firing? And there is one phenomena or period that we already learned before, that is called relative refractory period, and also absolute refractory period that takes about 1 to 5 to 10 millisecond. Okay, so in within 10 millisecond tu, there will be no over sensation or over firing of action potential. Dia mesti melalui all this refractory period in order to prevent overfiring, because overfiring of our nerve cell will cause distortion and also damage of nerve cell. Okay, so next, factors that affect the speed of nerve transmission. There were three factors: axon diameter, presence or absence of myelin sheath. So this is the aura. Okay, and also temperature. For axon diameter, larger diameter, for example, squid nerve cell or squid axon, there will be faster speed of transmission. Okay, faster speed of action potential transmission because less resistant. Okay, for squid axon, they do have larger axon diameter, but they don't have myelin sheath. Okay, kita smaller. For human being, we have smaller axon diameter, but we do have the myelin sheath. Our axon are myelinated. So to overcome the resistance of narrower diameter is actually being done by the presence of myelin sheath. Human being memang kecil je, dia punya nerve cell, tapi tak ada myelin sheath. Squid dia tidak ada myelin sheath tapi dia punya diameter axon dia besar, larger. So bila larger, less resistant. Bila less resistant, the transmission will be faster. And also temperature is the will explain the optimum temperature that can regulate optimum kinetic energy for whole metabolism in our nerve cell to make up more efficient metabolism or more efficient in nerve cells transmission of action potential. So this is the myelinated axon. So this is our axon. Okay, we are covered by Schwann cells. So this is the nucleus of the Schwann cells. So this Schwann cells is actually under glial cell. Okay, glial cell is not involved in transmission, but it is more for supporting and mechanical support. For example, secreting myelin sheath that can make the transmission of action potential will be faster. So this myelin sheath is actually secreted by this Schwann cell. Okay, myelin sheath consists mainly of phospholipid membranes of Schwann cell. Okay, dia ni adalah phospholipid membranes of Schwann cell, act as an electrical insulator and prevent the movement of ion across the myelin sheath. So dia sebagai insulator lah to prevent the leakage of ion across the axon myelin sheath. Okay. So this is the myelinated portion. Okay. This is the formation and also the maturation of or development of myelin sheath from the Schwann cell. You can see this is the bulk here is actually the nucleus of myelin sheath. So from here, will be project the secretion of myelin sheath cover up the axon membrane. So the part with no myelin sheath or the part with unmyelinated is called node of Ranvier. Okay, so dekat area without myelin sheath, this we can found sodium and potassium voltage gated channel. So the voltage gated channel can be found at this area at the nodes of Ranvier. So depolarization only happen at the node of Ranvier and not occur at the part with the myelinated sheath. Okay. Depolarization hanya berlaku di bahagian nodes of Ranvier yang tidak ada myelin sheath. So dia akan leap off lah from the area nodes of Ranvier here to the next nodes of Ranvier. Berlaku leaping of electrostatic. Okay, sebab dia takkan melalui depolarization takkan lalu di bahagian yang ada myelin sheath. So this is the detail. Okay, so these are the myelin sheath. This one, yeah. Okay, these are the myelin sheath. These are the nodes of Ranvier. Nodes of Ranvier here, and here. Okay, at this part, when the stimulus strikes, there will be trigger the sodium gated channel at this part to be open. So active node at peak of action potential means that once the sodium gated channel are triggered to open, sodium ion will diffuse in. When it diffuse in, you can see this is the depolarization occur. Berlaku depolarization. You can see starting -17 dia dah depolarize to +30. Okay, di kawasan ni still negative charge because at this time this area is under inactive state. So adjacent inactive node into which depolarization is spreading will soon reach threshold. Will soon reach threshold ni kenapa? Because this is negative, this is positive. There will be local current flow. Okay, positive will be attracted to the negative. Am I right? So positive will be moved to this area and will form local current flow. So this local current flow will then depolarize this area. Will depolarize this adjacent inactive node from resting to threshold. You can see from resting -70 to threshold. Okay, so dari sini mula-mula dia ni this area depolarize. Okay, there will be accumulation of sodium ion. Increasing sodium ion repels the cation to the adjacent node of Ranvier, which is more negatively charged. So, a local electric current flow is set up jika ada tarikan from positive to negative. The local current causes the depolarization of the adjacent node. The threshold level is reached. Okay. The action potential produced at the second node of Ranvier then generate a local electric current that depolarize the third node of Ranvier. This is the first node, second node, third node. Once this area already being reached the threshold level, okay, the second node of Ranvier then will generate a local current that will polarize to the third node of Ranvier here. Bila sini dah jadi positif sebab ini tertarik ke sini, ini positif. Later, ini positif kan? At the same time, the remainder of node is still at resting point. Semasa ni di negatif, ini positif, tertarik ke this region. So another local current flow set up. Okay, I come again. Mula-mula yang ni depolarize. Ini negatif. Positif negatif akan attract. So dia akan pergi ke sini. Leaping. Dia akan leap. Leap to this. So bila sini akan terkumpul positif ion. Di sini still lagi negatif. Then once dia dah capai threshold level from the second node, later will generate local electric current that depolarize the third node of Ranvier. The action potential therefore leaps. The action potential therefore leaps from one node to another increasing the speed transmission by up to 100 times. So leaping progress ni will speed up the transmission of action potential. So this phenomena we call as saltatory conduction. What's the significance of this saltatory conduction? It will increase the conduction of velocity without the need to increase the axon diameter. Without increasing the axon diameter, we still can perform speedy transmission due to this saltatory conduction without consuming more energy. So less energy is needed for conduction because less surface area is involved and fewer ions, either sodium and potassium ion, has to be transported across the membrane. Sebab dia macam tarikan elektrik from this point to this point, and then another local current set up from this point to this point. So tidak perlulah all the sodium ion nak kena masuk di setiap area of the exoplasma. Kan? Only area at node of Ranvier je yang terlibat. Kawasan yang ada Malaysia tak terlibat. So dari situ ada economical value. Tidak perlu ruang yang besar dan tidak perlu menggunakan sodium potassium pump dengan banyak, and also tidak memerlukan lots of sodium and potassium ions diffuse in and out, and this can save the ATP consumption. Okay, that's it. Thank you. Okay, before that you can see here, ni direction of propagation of action potential. Nampak tak ni? Starting from here to this area. Ni direction of propagation. Maknanya why we use propagation? Because the action potential is kind of an electrical wave. Because when we talk about wave, the term that's suitable for them is propagate. Means that's what we call as a propagation of action potential. Okay, that's it. Thank you.